Hello, I'm going to hit presentation mode and continue the lecture from this point. Okay, so when we last left off, we had talked about sensation and perception and what they were. So as a quick review, sensation is the bottom-up puzzle piece process that allows us to take individual pieces of information and transduce or convert them through special receptor cells in the sensory systems and turn them into a brain signal or neural impulse, which is action potentials and, of course, the chemicals that go to the brain. When the brain gets those chemicals, then it can interpret what we are experiencing in the real world. So, for the visual system, which is what we were concentrating on right now, then we will have our stimulus, which is light waves. In the auditory system, we have sound waves. So we have both visual and auditory systems have the same physical stimulus, which is a wave form. However, as we talked about, light waves will only work in the eyes, and sound waves will only work in the ears. So as we are going through this today, or whenever you're doing this, the visual system will be first, and then we'll talk about the auditory system in very similar ways with the other senses next time. So the terms that we're talking about now are wavelength, hue, and intensity, although I will also go over what this would look like for the auditory system. So, wavelength. The wavelength is the distance from the peak of one wave to the peak of the next wave. You can have a few waves if they're farther apart, or you can have more waves if they are closer together. So, these electromagnetic waves vary from short little blips of cosmic rays to the long pulses of radio transmission. So very much like the ideas of sensation and perception being on the same spectrum, right, part of the same process, radio waves or audio waves and visual waves, even though they are two different types of waves, are still along the same spectrum. So radio waves and TV waves, basically, and things like that. So the shorter the wave, the more of a more of a blue or purple color you will see. The longer the waves, the farther the distance apart between waves, the more of a red color you'll see. So the dimension of the color determined by wavelength is called the hue. These are what we know as the color names: blue, green, red, yellow, and so forth. That is determined by the distance between the two peaks of the wave, or how many peaks over a given amount of time. I'll show you what that means. We call that frequency. And then we have the height of an individual wave itself. The height of an individual wave is known as its intensity. We call this the amplitude or height of the wave. So it's not the distance from peak to peak, it's the distance or height of one wave from the bottom of the wave to the top of that wave. And that is a matter of intensity. For light, that tells us how bright the light is, or if it's a smaller wave, how dim the light is, how bright a color is, or how dim the color is. And in the auditory system, that would be the loudness of the wave. So for the auditory system, the intensity would be a higher wave would be a much louder sound, and a smaller wave in height would be a much quieter sound. What about wavelength in the auditory system? Well, in the visual system, wavelength is determined by, determines the type of color that we see. In the auditory system, we also use wavelength, and that determines how high or low the pitch is that you hear. So what that means is, much like the color red, which is far apart waves, as you'll see on the next slide, the farther apart the sound waves are, the lower the pitch will be. The closer together that the waves are, the higher the pitch will be. I know, I'm not very good at that. That's okay. Thank goodness you don't have to see me. So if we're looking at wavelengths, we can see that the waves vary in their distance between the peaks. So here you see in a blue color that the waves, or the peaks of the waves, are closer together than below it in red. Here we see the amount of waves that pass over a given time. So sometimes a wave is called a cycle, and we can look at how many cycles we see in 1 second. So if we were to look at the picture on the left and look at the blue, you can see the arrows, which are shorter than the red, from peak to peak. In this time, if we were to say number of cycles per second, what we see is 4 peaks in blue, and we only see 2 in red. Those red wavelengths have a lower frequency because the distance between the peaks is longer. So in the same amount of time that 4 blue wavelengths, or 4 blue peaks, pass through, only 2 red ones are. And that is sort of the math that our brain does that lets it know what color we perceive. So shorter, faster, higher frequency is going to be more of a blue or purple, and the longer the frequency, the more red you're going to see. Green, yellow, brown, all those other colors, kind of somewhere in between those. So waves vary in their wavelengths, which is the distance between the peaks, and we measure it by frequency. So the number of wavelengths that pass in a given point of time, and that depends on the wavelength. So again, as you see, 4 blue at the same time as you'd only see 2 that are red. Waves also vary in their amplitude, as I said, which is the height. So you can see on the bottom that the bigger the height from the bottom to the top of the wave, only 1 peak, only 1 cycle, right, 1 wave, is its amplitude or intensity. So it's the height from the bottom of the wave to the top of the wave. So the yellow one, with a higher amplitude, higher wave, is going to be brighter. As you can see from the slide, that the green one has a pretty small wave. So the color itself of green is pretty dull. It's not a nice, sharp green. It's kind of a dull green because the wave is smaller. So again, if we were to do this for the auditory system, what we see is that the blue would be faster waves than the red. So in this case, the blue would be a higher sound, and the red, being very low frequency, the peaks are far apart, there's a longer wavelength, that would be a lower sound. If we were looking at it in amplitude, again, it determines not just the brightness of colors, but in sounds it would be loudest. So the louder sound would be the yellow wave, and the quieter sound would be the green wave, the small amplitude. So just to review, in the visual system, which is what we're going to continue to talk about, shorter wavelengths are more of a bluish color, longer wavelengths are more of a red color, and the others are kind of in between those wavelengths. So the wavelength determines basically what color or hue that we perceive in our brain. And the amplitude or height of the wave tells us how bright or dull the color is. So now when we look at the eye, we can see the outline of the eye. We are looking at the eye as a whole, but we will be focusing on that area in the back of the eye that's called the retina as we go through. So first, what are we seeing here? Well, we're seeing a person with a brown eye, and we're looking at the diagram of the eye to see what's going on. The eye is focused on, as you can see, a candle. The candle is lit, looks like a birthday candle in the visual field. And what you see is that that image is projected onto the back of the eye in the retina in an area called the fovea. As you can see, the fovea is the point of central focus. And what we see is that when this picture or image is projected to the back of the eye, it is upside down. What you can't see is it's also backwards, like a mirror image. But the interesting thing about that is, even though the picture is projected upside down and backwards, when that information gets to the brain, what it does is it re-inverts that image again, so we see it just as it is in the real world. And if you ever did see old cameras that had actual film instead of digital, when you looked at what we call the negatives of that, you saw something similar, that the pictures did not really have color and that they were kind of backwards. And when they were developed, when the film was developed, the picture comes out the right way as it was taken. So let's review the parts of the eye, and then we can go through with how this information goes from the eye, or from the real world through the eye, these light waves, and how they make it to the brain. So the first thing we see is the outer layer of the eye called the cornea, and you can see the word cornea right below iris. The cornea is just an outer sort of layer that protects the eye. And I don't know if anybody's ever scratched their cornea, but I have. It was a dodgeball accident where somebody threw the ball from the opposite corner and I didn't see it. Now, normally when our eye senses that there is danger coming, we blink to keep out dirt and things like that, or to protect our eyes. I did not have that luxury, and as a result, I had a scratched cornea, and I could not play dodgeball basically for the rest of the season. Towards the end, a little bit, but I had to wear these silly goggles, and we don't need to get into that. So the cornea protects the eye. It's just the outer layer. What it's protecting just inside the cornea is the pupil and the iris. So if you look at the picture of the person's eye just to the left, that big black part in the very center of the eye is called the pupil. Does anybody know what the pupil's for? Well, since I don't have an audience live in front of me, I'll tell you. The pupil allows light to enter into the eye. And some of you guys may notice sometimes that people's pupils get bigger or smaller depending on what's going on. In the dark, pupils get bigger because they need more light information to see basically anything. But as we talked about in the last lecture, if you're going outside and it's really bright and you just came out of a movie, or maybe now out of your dark house into the bright light, your pupils are going to get much smaller because there's an awful lot of light that's already entering, and we don't want to overload our senses, especially the visual sense. So the pupil gets smaller and only allows a certain amount of light in that we need in order to be able to navigate our world. So the pupil gets bigger and smaller in response to the amount of light that comes through. Now, around that pupil is this thing called an iris, and the iris is the colored part of your eye. So if you see that picture on the left, it's the brown of this person's brown eye, like myself. The iris is actually controlling the pupil. It is making it bigger or smaller. It's expanding or contracting the pupil based on how much light is needed to enter into the eye. So although you may not have thought that the colored part of your eye played a role other than just being a certain color and making you look handsome or pretty or whatever, it's actually controlling the pupil and expanding and contracting the muscle. So the iris is controlling how much light gets through the pupil by making the pupil bigger or smaller. Once light goes through the pupil, as it's controlled by the iris, then just like a camera, it hits that part right behind it called the lens, just like the lens of your phone camera or your digital camera or any video equipment that you may have. The lens focuses the picture and projects the image from the real world through these light waves onto the back of the eye, which we call the retina. So some of us can't focus on our own our lens, right? Genetic conditions, or as you get older. And so some of us have corrected lenses like glasses or contact lenses where we put over our eyes, and what it does is it refracts the picture and makes the lens project the image a lot clearer because we don't have that luxury ourselves. If you don't wear glasses or don't need them, it's because your lens is just fine on its own. So the picture is sent to the back of the eye and projected on the back of the eye upside down and backwards in the retina. And as you can see, the retina is that whole layer in the back of the eye. But there are two specific parts on the retina that need to be pointed out. The retina goes from the top of the eye in the back all the way around, and you can see the sort of little layers of cells. The two parts that we really want to focus on are the fovea, and that was a bad attempt at a joke because that is the point of central focus. It's in the center of the retina. And that is where the picture is going to be focused. There are certain cells called cones, which are concentrated mostly in the fovea. 95 to 99% of those cones are in the fovea, and those are the cells that allow us to see things really clearly. The other part of the retina that you may notice right below the fovea is an area called the blind spot. And why would there be a blind spot in the back of the eye? Well, as I said, the information needs to get from cells in the back of the eye to the brain. So, as we talked about in chapter 2, the information in the cells gets transferred down through axons. And there are certain cells whose axons come together to form what we call a nerve. A nerve is a bundle of axons that are kind of formed together. And where that information is leaving the eye and heading to the brain is called the blind spot because it's a bunch of axons. It's not cell bodies. So you can't project anything there because that's the point where the information is leaving the eye and heading to the brain. We call that the optic nerve, sending visual information from the eye to the brain. And where those axons meet and leave the eye is called the blind spot. Now, if we were to zoom in, as you can see, onto the back of the retina, you see that there are 3 layers of cells. And although the light would seem to hit the first set of cells first, it is that back layer on the very back of the retina in the eye, very back of the eye, that actually causes the signals to transduce into a signal the brain can understand. So, even though you would think it would go 1, 2, 3, because it's not activated till it hits the back layer, that's the first layer of cells, and then it kind of goes forward from there. So it's a little bit the opposite of what you might think. When light hits the eye, now we're going to talk about how it turns into a signal and which cells are involved. When light hits the eye, it hits that back layer of cells. There are 2 types of cells named, again, for what they look like. Those are rods and cones. The green ones are rods because they look like rods, and the orange ones kind of look like traffic cones. At least that's what they look like to the first person or people that discovered them under a microscope. So these rods and cones together are called the photoreceptors. Rods and cones are both types of photoreceptors, and they give different types of information to the brain. So before we get into that, just know that when light hits the back of the eye in those rods and cones, the first layer of cells, that in response to light, the cells give off a chemical. So light hits the rods, light hits the cones, and in response, the cones give off a chemical. And now, as we talked about in chapter 2, the chemical is picked up by the next cell. So the chemicals that are given off by the rods and cones are picked up by the cells in layer 2 called the bipolar cells. And the bipolar cells basically are just giving the information from the first layer to the third layer. So bipolar cells are kind of the messengers that take the message, the chemicals, from the rods and cones and give them to the ganglion cells. So again, that makes the photoreceptors what we call the receptor cells. Remember, those are the ones that transduce the signal. So rods and cones turn light waves into a chemical. That chemical is given to the bipolar cells, and the bipolar cells then give that same chemical to the ganglion cells. That's the third layer. The ganglion cells are the ones whose axons come together, as you can see in the picture. The ganglion cell axons are coming together to form the optic nerve, and that leaves the eye through the blind spot. So again, to try to make it clear as possible, light comes through the eye, through the lens, projects onto the retina. When light hits the rods and cones, also called the photoreceptors, those rods and cones give off a chemical in response to the light. The signal is now transduced. It has now been turned into a brain signal via chemicals and action potentials, and it is no longer light waves. So now the brain can understand this. From the rods and cones, that chemical activates the bipolar cells, action potential, and then those chemicals are given to the ganglion cells. So the rods and cones send their information through the bipolar cells to the ganglion cells, and it is the axons of the ganglion cells that send that information from the eye to the brain. When they do that, it goes down the axons of the ganglion cells, which come together to form the optic nerve, and the optic nerve leaves out of the blind spot, because it's axons, not cells, as it goes to the brain out the back of the eye through the optic nerve. So what are these rods and cones? Well, again, the green ones in this picture look like rods, and they are more numerous. And in this one, we see blue types of cones, which look very coney. The way to figure this out is to understand what each of them does. So the rods are very numerous. There's a lot more rods than there are cones in the visual system. And these rods have very small, what we call visual fields. So they're giving information to the ganglion cells, but since there's so many of them projecting onto each ganglion cell, they only give a little bit of information. So they work really well in low light when it's sort of a blurry, fuzzy picture. It's all very black and white, and they can help you see sort of the outline of shapes or things, and when something's moving. Because when you see something moving, the background is still, but the thing, the object, the person, whatever it is, you can tell that they're moving while the background stays still. So that is the rods that really go into the house, you're not turning on any lights, and it's very dark in there, and all you see is maybe the outline of like the couch and the coffee table, and you're just trying to not hit your shin on the coffee table and hurt yourself and make too much noise because you're sneaking in. That's your rods. The rods are doing all the work there. The cones, on the other hand, are what I like to call the 3 C's. So cones equals color, cones equals clarity or clear vision, and cones equals center because they're located in the center of the retina, or what we know as the fovea. But a lot of times it's easier to remember when you use what we call a mnemonic device, where you sort of make a mental picture or some way of trying to remember the information better. So that's why I give you this. Cones equals the 3 C's. Cones are responsible for the color portion of your vision, whereas rods are black and white. So cones equals color. Cones send the information about the colors that you're seeing with those different wavelengths or frequencies to your brain to interpret. Cones are equals clarity. The rods are very black and white, fuzzy picture. So the cones are really giving a crisp, clear picture to it. And that brings us to the third C. Cones are located, 95 to 99% of them are located in the fovea, which is the center of the retina. So that is where the information is clear. Why is it so much clearer? Why do we get vivid colors through the cones? Well, remember, there's very little cones as opposed to rods. So the cones are basically a one-to-one connection with those ganglion cells. And because of that, everything the cone has, it gets projected to one ganglion cell. So the information sharing is very easy and very direct. Whereas you have 100 to 1,000 rods going to one ganglion cell. So you're getting very, very little information from each individual rod. So rods are fuzzy, black and white, not very detailed. You can see black and white, you can see in the dark, you can see the shapes or outlines of things, and you can see movement because of the rods. The cones are the 3 C's. Cones equals color, cones equals clarity or clear vision, and cones are located in the center of the retina in that area we call the fovea. So if I go back really quick, a couple slides, here you can see the fovea is the point of central focus. That's where majority of the cones are. The rods are going to be in the retina all around on the periphery. So cones equals color, cones equals clarity, cones equals center of the retina or central focus in the fovea. Now, you may not realize that you already know half of this information from chapter 2. I'm just putting the pieces together for you. So, we already know that 4 of the 5 senses go through an area of the brain that we have discussed previously. That was on your exam. And from there, it's really only one other step. So, let's look at the slide first. How does the brain turn light stimuli into useful information about the world? It collects all of those frequencies and intensities and things like that, and it collects and analyzes this information. We call it sensory, right? The 5 senses. And how it does that is converting them into a neural signal, into a brain signal. So those are the chemicals and the action potentials. And the information that is transferred goes through the optic nerve and to the brain. So you already know half the information. Once the information leaves through the optic nerves, as you see out the back of the eye, it heads to the brain, and what you see is that it crosses over before it goes to that area of the brain we call the thalamus. So the point where the axons cross over is called the optic chiasm, Chiasm, chiasm. And that comes from the Greek letter chi, which is an X. Optic chiasm is just the point where the fibers or axons of the optic nerve cross over, and it makes an X, as you can see. What you can also see, if you follow the color pattern, is that not all of the axons are actually crossing over. In pink, on the right-hand side, what you see is that some of the axons stay on the same side, where in the left eye, in pink, it's crossing over. Same thing with the right eye. On the right eye, you can see the black axons crossing over. In the left eye, the black axons are staying on the same side. So in this case, as we talked about in chapter 2, we know that about 85 to 90% of the axons in the sensory system, and this is for all 5 senses, not just vision, crosses over. So we would say that majority of them cross over. So that's not an actual, stop on the tour, as they're still crossing over. The first stop, or we call it the synapse, where it's connecting to other neurons, as you can see, right? Remember the term synapse was like the gap between 2 neurons. Where the information from the optic nerve first synapses or first stops is, you guessed it, in the thalamus, because 4 of the 5 senses, except for the sense of smell, goes through the thalamus. Remember that the thalamus is our relay station and our amplifier. So even if the signal from the eyes was kind of weak, the thalamus makes sure it gets to where it needs to go, and the thalamus makes sure that the signal is as strong as it can be. It amplifies the signal. From the thalamus, there's only one more stop on this pathway. So it seems intricate, but it's really not that difficult. From the back of the eye, It crosses over in the optic chiasm or stays on the same side, and then it goes to the thalamus. From the thalamus, that's only one more stop. It goes to the visual cortex, which is located in the occipital lobe. And again, in chapter 2, we talked about that the occipital lobe only has one function: visual processing. So from the thalamus, the information goes to the visual cortex. That's it. It goes and gets transduced, right? If I were to go back really quick, from the rods and cones, light hits them in the back of the retina. That information, through chemicals now, it goes to the bipolar cells, and that information is given to the ganglion cells in layer 3. The ganglion cell axons form the optic nerve that leaves out the blind spot of the eye. From that spot, as you can see behind the eyes, the optic nerve, about 85 to 90% of those axons cross over. So majority of the information from the left eye goes to the right thalamus. Majority of the information from the right eye goes to the left thalamus. And from the thalamus, the information goes to the visual cortex so that the brain can interpret what it is the eyes are seeing. Now, we talked about black and white and we talked about color. There were two theories of how color is processed. One theory is called the Young-Helmholtz trichromatic theory. And Young and Helmholtz were two researchers that noticed, and we found that the retina has 3 types of cones. It has a red cone, a green cone, and a blue cone. And the combination of those cones respond to the different color stimuli that makes up the other colors that we can perceive. So, in this case, the stimulation combinations, wavelengths and frequencies and brightness, intensity, all of that stuff, is a combination of these 3 main ones: red, green, and blue, or RGB. If you've ever seen the back of a TV or a projector or things like that, there is an input that's marked RGB, and that RGB stands for red, green, blue, which corresponds to our cones. But one thing you may notice if you're a fan of painting, or since you've been exposed to it since pretty much kindergarten, you probably know that red, green, and blue are not the primary colors that you learned in art. Those colors are red, blue, and yellow. Because if you recall, and if not, I can remind you, that yellow and green make blue. So why are red, green, and blue the primary colors here? Well, we don't really know why that is, but what that tells us is that this trichromatic theory is an incomplete theory, because if your primary colors are red, green, and blue, how do we even process the color yellow? We can't in this, according to this theory. So this theory is correct about the RGB, about the red, green, and blue cones, but it doesn't account for the color yellow. So therefore, it is an incomplete theory by itself. Another theory was theorized, I guess is the word. Another theory was that somebody else came up with a few years later, was a man named Herring, and he called it the opponent process theory. What he said was, it's not the cones necessarily that are the reason we see the different colors. But remember, what are the rods and cones attached to? What do they send their information to that actually goes to the brain? Those are the ganglion cells of layer 3. Those are the axons that form the optic nerve, and that's the information that goes to the brain. So what Herring said is the way that information is taken by the ganglion cells from the cones is in what we call an opponent process way. So if they hit one part of the ganglion cell, they'll see one color. And if they hit another part of the ganglion cell, they'll see the opponent color or the opposite color. And if anybody knows the 2 main types of color blindness, the main types of cells as far as ganglion cells are red and green, and the other one is yellow and blue. And of course, if we add the rods in there, black and white are also opposite processes. So what Herring said is it's the way that these ganglion cells receive information. If you hit a certain part of the cell that are red-green, then you're going to see red. If you hit the other part, you're going to see green. And the same thing with yellow and blue. Now, this brings up a very interesting thing, because there was this thing that broke the internet a few years ago, the original thing that broke the internet. And that was, if you recall, the dress. And the dress was either seen as, that's right, black and blue, or, based on this theory, what is the opposite of that? Yellow and white, or white and gold. So people either saw a dress that was black and blue, or they saw a dress that was white and gold. And the only thing that they were sure of for sure is that what they saw was correct. Now, before we get into the solving that little riddle, it turns out that the best way to process colors is a combination of both of these stages. This is the take-home message. The Young-Helmholtz trichromatic theory, which is the RGB, the red, green, and blue cones, is correct. We do have a combination of red, green, and blue, but it by itself is incomplete because it doesn't account for the color yellow. Where yellow comes in, and all the other colors, is these red, green, and blue cones and how they project to the ganglion cells and how the ganglion cells take up that information, which is that opponent process fashion. That's why some people with varying degrees of, right, their absolute thresholds and difference thresholds and things like that, remember we talked about that last time, is that it varies from person to person, how much you're paying attention, how tired you are, blah, blah, blah. So some people's sensitivity in their cones and their ganglion cells is going to see one, and some is going to see the other. So you either saw the dress as black and blue, or its opponent process, white and gold. That's why the two types of color blindness are red-green, because they're opponent processes, and yellow-blue, or gold-blue, because they're opponent processes. That doesn't mean we can't see them together, but those people who are colorblind, if it's red-green color blindness, they can't tell the difference between any shades of red and green, and for blue and gold, or blue and yellow, it's the same thing, but for the colors blue and yellow. And of course, black and white are opposites. So in the video, that's the 10 mind-blowing illusions. The dress, of course, is considered in those, and you will see that it is either yellow and white and gold, or it's black and blue. And so the answer is as to who's right. Remember back to that first lecture where I said that perception is reality. So if you see it as white and gold, then it's white and gold to you, because your perception is your reality. If you saw it as black and blue, then it's black and blue to you, because your perception is your reality, right? Hopefully that takes care of that, although debates are always fun. So the rods then are involved in this feature detection, as I talked about, where we can see things such as shape, angle, movement. Those are more the rods. Color processing, making sure the picture is nice and clear, that's the cones. But when we look at the back of the brain, that's the occipital lobe that's all lit up with different colors. There were two scientists named Hubel and Wiesel, and you don't need to know their names specifically for the next exam, but they basically shows that what we talked about with sensation and perception. The brain deconstructs, right? The neurons deconstruct the image. They bring the individual puzzle pieces and then reassemble them once they hit the brain. That's the top-down processing. And what Hubel and Wiesel found, and this is sort of new technology built on their old one, because they did this in like the 1920s or 30s, is that there are special clusters of neurons in the occipital lobe, in the visual cortex, or cortices, that receives information from the ganglion cells. And that when we're trying to put things together, one of the things our brain is doing is putting shapes and objects and things together to make the full picture. So as you can see from here, there are special clusters of neurons at the very bottom in green that when people were looking at houses and they were recording cells, those are the cells that basically had action potentials and did their thing. When they were looking at chairs, it was these clusters of cells in the color blue. Looking at faces, it's those two little areas, the bigger one above the green and then a little tiny one above the blue that recognize faces. And if you put things together like houses and chairs, a little higher up in the brain, you see it putting it together. We kind of call that higher order processing. The more complete the picture becomes, or the interpretation, the higher in the brain it gets processed. And as we talked about with the frontal lobe, right, putting all this information together from chapter 6 and chapter 2, once all of those visual images and things are put together, that information travels to the frontal lobe where it integrates all the other senses, right? So what this also means is if those cells, those neurons were damaged, the ones just in red, you would not be able to recognize people's faces, even if it was your mother that you saw every day of your life. If you got into some accident on both sides of the brain and those areas in red were damaged, you would be able to, see things and see shapes and stuff like that, but you would not be able to recognize people's faces. So you went home and you looked and your mother answered the door but didn't say anything, and you're like, I don't know who you are. But if you closed your eyes or if you just heard her speak, you would say, oh, that's mom. You just wouldn't be able to recognize her face because those neurons are specific in recognizing faces. So that means that different neurons in different parts of the visual system, even in the same area of the brain, have different things. When we are putting the picture together, right, we're taking those individual puzzle pieces and putting them together to make the whole story. That means that different clusters of neuron within the visual system are focused on or are processing different things. Certain neurons are processing the color, certain neurons, right, and that would be from the cones, information from the cones. From the rods, we're getting form and motion, right, and clarity and depth perception would also be part of the cones. So we are getting information from the different types of photoreceptor and different clusters of neurons that are processing things like motion, form, depth, and color. And even though they're coming up individually, remember that our brain is putting things together at the same time in the big picture. That's our perception. So we know that this happens within milliseconds, and that even though we get the puzzle pieces coming at different intensities and wavelengths and all of that, we're putting the picture together almost instantly once it hits the brain from those different areas of the brain that are processing things, right? And we know that this is true because of patients with brain damage, kind of like we talked about with Phineas Gage in chapter 2. Studies of patients with brain damage show that if you damage certain neurons or clusters, that you may not see faces, or you may not get motion or depth or color of any sort. And what that tells us is that the brain takes this deconstructed image and it puts the big picture together simultaneously. So the fact that we have different neurons working on different aspects of our vision and putting together the picture at about the same time, we call this parallel processing, because those parallel clusters of neurons are processing information and putting it together in the big picture at the same time. So we call that parallel processing. Now, the next question that arises from this is, how do we see different shapes, colors, and things that are put together? How do we see it as one thing? Instead of looking at rectangles and lines and things like that, I see a desk. Instead of looking at black and white contrast in this big square, I would see a projector screen, right? How do we know what something is and that it's not just all the things it's made up of? Well, there is a type of basically subfield of psychology that was started in the 1950s or 60s in Germany called Gestalt psychology. And Gestalt basically means the whole is greater than the sum of its parts. So Gestalt psychologists wanted to determine just that question. How do we take a bunch of shapes and put them together to make sense into what we see in the world? So Gestalt psychologists propose certain principles that are used to organize these senses into a whole perception. And that includes the form that we see, how far away it is from us, depth, and the most important part of all, basically, which kind of ties into sensory adaptation, is perceptual constancy. If I was lecturing to you right now and I turned from facing you to sideways, facing the door, how do you know that I'm the same person? When I go in front of the projector screen and it's really bright, my clothes don't change color to you. You still see the same colors that I was wearing. When I go behind the desk and you can't see my legs, how do you know that I'm still a full 100% me? You know because of what we call perceptual constancy. That's this idea that even if we change shape, get closer or farther away from you, so change your depth perception, or if I, lean over or go into a brighter light or darker area, that I still seem the same person, because we don't want to trick our brain into thinking that I'm somebody else, right? That would be really hard to navigate the world. If we saw a bird fly right across your visual field, when it turns and flaps its wings and goes into brighter and darker areas, shaded or sunny, you see that it's the same bird the whole time. And that allows you to know what's going on in your world, right? That lets you know when lights change to drive. That lets you know when people are walking across the street. And the fact that we have constancy doesn't freak us out and make us think something different is happening, because how would you live in a world like that? You wouldn't survive very well. With sensory adaptation, you wouldn't be able to adjust, or your cells would get tired because they couldn't adjust. With perceptual constancy, it's sort of the same idea, that it allows us to understand what it is that we're seeing or hearing or feeling, blah, 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 and that it doesn't change on us. So we can navigate through our world and, of course, survive. So how do we organize shapes into colors and meaningful perceptions? Well, our brain tends to fill in the blanks. Here we see a bunch of circles that are incomplete because there's some parts of them that are missing. But if we were to concentrate on that little X, right, in the second row of circles from the top on the left, there's a little X. If you were to concentrate your attention on the X, your brain basically tries to fill in the blanks or turn a bunch of pieces into something whole. And what you get is an organized gestalt. You get a whole picture. In this case, what we're seeing is a three-dimensional cube filling in the spaces of the circles, even though it's a two-dimensional object. You're seeing it on a computer screen, which is two-dimensional. But you're seeing a three-dimensional picture because your brain is filling in the blanks. This is how that works, right? So we are taking things and putting the shapes together into something meaningful to us, and that includes our schemas from the last lecture. So how do we know where one object begins and another ends? Well, one of the things we did in the contextual piece was look at the difference between the foreground and the background, or what is known as figure ground. Even though humans in the eastern and western side of the world do it differently, on the eastern side they tend to see the background more, and then the foreground or figure. In the western part of the world where we are, we see the figure or foreground first and then the background, and that allows us to make that connection between what's going on and what's not. And again, our interpretation, are they inside or outside, is based on your perception. We also tend to group things together that are very similar, and that helps us to understand even things we've never come into contact with, sort of what's going on with them. The three main ways, and this is important, so please pay attention to this slide, the three main ways that we group things together are based on these three principles: proximity, or how close the lines are together; closure, whether or not the shapes have an open or closed end to them; and continuity, whether they are flowing sort of together. So with proximity, what we see here are 6 lines. But those 6 lines, we would tend to say 3 columns as opposed to 6 lines. Why? Because 2 lines, 2 lines, and 2 lines are very close to each other. So although there are 6 lines there, we tend to see, if someone were to ask us, what do we see here? We would tend to say 3 columns because of the lines that are closer to each other, like 3 freeway lines or something like that. In the second one, where we see these little circles that are not filled in between the circles, you can see in white, right? Your eyes will fixate on what's not actually there, which is the shape of a triangle. But if you notice between the left and the right circles, that the triangle on the left seems to be brighter or more vivid. And that is because the little circles don't have any lines closing them. So it seems like the object, the triangle, is connecting the 3 shapes. On the right, you see that they're closed, so they kind of look like C's. And so you can still kind of faintly see the triangle in there, but it is not nearly as vivid because the shapes are closed off. So if the shapes are closed, we tend to see them more individually. If they're not closed, that's when we see something in the middle. And in continuity, what we see is a line that's going through what looks like it could be a light wave or a sound wave, since I use those shapes for this lecture. Because they are occurring together, it seems like they're integrated. In this case, it looks like the line is going in front of, behind, in front of, or the opposite, behind, in front of, behind the wave. It looks like, instead of being 2 different things, a line and a wave, it looks like they're interacting together. So that is how our brain interprets these things, right? Proximity, how close together the lines are, whether the shapes are open or closed together, as to whether we see something in the middle. Our brain tries to fill in the blanks, and continuity. So this idea of perceptual constancy is, no matter how close or far away we get, remember, our brain is basically doing math, right? It is interpreting wavelength, it's interpreting frequency, it's interpreting amplitude or height. It's interpreting how many action potentials we have, because remember, it's more action potentials, not the size of an action potential. So our brain is doing math on these things. And so even though we see different equations because we are closer or farther away, because things are turned a different angle, they have a different shape, different amounts of illumination, amounts of light, changes in the retina, we still see colors, size, and depth as constant. And that, again, allows us to navigate our world and not be freaked out by the smallest thing. We can see that things don't change because our brain can allow us to survive our environment and continue to do the things that we do, to drive home, to get out of people's way if we need to do what it is that we do. One last sort of question about the visual system is, okay, we know that when children are born, that they can see, we know that they can cry, we know that they can feel somebody holding them, but are those neurons and connections to the brain, like we'll go back to the visual system, are those neurons, those ganglion cells, connected to the thalamus, connected to the visual cortex or occipital lobe, right? Visual cortex in the occipital lobe. Are those strong enough at birth because they have fully formed before we were born? Or do we need to continue to get visual information to sort of update and improve upon the pathways? And researchers have been wondering this for a long time, or at least they had until they actually did this. Now, back in the days, 1910s to 1950s and things like that, there were no restrictions on who you could test. So what they did was they did what we call sensory restriction, where they basically took away visual information from these infant animals and humans. So cats, monkeys, humans. And what they did was, after they were born, they basically put them in a dark room with no visual information.
or you can, if it's a human, you could put, a blindfold on them. And so what they wanted to see was, if we basically took away their visual information for the first few months of life, would it be fine because they had it already, the system in place before birth, or does it need constant stimulation to sort of update the network? And what we found was, well, not we, what they found was, is that those animals and babies that were basically left in the dark and not allowed those neurons to develop became effectively blind. And so what this tells us is that there is a critical period for normal development, a critical period of development in the first, we'll say, 6 months of life, that without stimulation, these connections do not develop. So even though we can see at birth, we need to continue to be able to see to make these neurons better, to make the networks better, the connections better, and to allow us to be able to see. And if they don't develop normally, we're going to lose that, right? So based on the connection from the eyes to the brain, you may think that people that are blind are only blind because of problems with their eyes. Well, now that we know the connection between the eyes and the brain, with the ganglion cells and the thalamus and all of that, we know that if that connection, that pattern, that pathway from the back of the eye to the brain is interrupted, or if there's damage to the part of the brain where the information goes, that can cause blindness as well. And of course, we have our adaptation, not just if I go back, not just perceptual constancy, we also have the ability to adapt. And I know I'm going to make some of you guys sad, because we've all seen those posts on social media that only 3% of people can replace numbers with letters, or adjust to a displaced or inverted visual field. If we see things backwards or mirror image or upside down, we can't really read the letters. Well, that's not true, because the vast majority of us, it's probably only about a small percentage that can't do this. So if I was replaced with the number 1, or an E was replaced with the number 3, or things like that, we are still available. We're still able to adjust and actually be able to read that. So I apologize if you think you're the top 3% of the world because you can read an I as the number 1, but it's just not true. So again, just like horoscopes, I'm sorry to ruin your hopes and dreams. I hope you've enjoyed this lesson. And when we come back for the next one, we will talk about the non-visual senses, starting with hearing, and I will recall and review the similarities between the visual system and auditory system, and we'll talk about the others. It's a much shorter lecture, but hopefully you'll enjoy. So thank you very much, and I am going to stop recording.